Austria for working on food authenticity. So last week I was there, and in fact when Dr. Bansal called me, this idea clicked me there only because we are working on food authenticity using isotopic fingerprints. So I found my project partners from all over the world. There were 22 countries. So many are working on different natural products. So I thought, why not to give this idea that, I mean, it may be new or may not be new, I do not know, but isotopic fingerprints, they are finding quite a good place in food authenticity, and I think they are finding place in authenticity of natural products also. So coming to natural products and the ingredients, we see that this market has grown a lot since last 3-4 years. During COVID time, it has really jumped because people have become more health conscious and more towards the natural products. But at the same time, I mean, there are two types of such nutraceuticals, I would say, or the health supplements. They are natural as well as synthetic. For example, we see vitamin C from citrus foods also. We see vitamin C which is chemically produced also. But these natural products, they are in high demand, but how to distinguish that whether these ingredients, they are from natural sources or they are from synthetic sources? Because we know that naturally produced things, they are a bit expensive as compared to the chemical production houses, because in chemically produced, we can produce in bulk. So unfortunately, because of demand as well as because of the global market which is growing, the high value labeling claims, they are getting, now I would not say it is adulteration. When we replace the whole product, it is not called as adulteration. It is actually we can say fraud, or it is the authenticity issue. So now how to distinguish the natural products and the synthetic products? I know there are other techniques also, but this is one of the niche techniques which is finding application nowadays. So isotopic fingerprinting, because based on the photosynthetic pathway, based on the origin, based on the geographical location, different products, they have different isotopic signatures. So this basis can be used for distinguishing between natural and synthetic, and for distinguishing the geographical origin. So major stable carbon isotopes which are being used for this purpose, they are carbon, hydrogen, oxygen, nitrogen. For example, carbon is being used for testing the authenticity fingerprinting. Hydrogen and oxygen, because they come from water and from carbon dioxide, so they tell us about the origin, geographical origin. For example, we produce here in Darjeeling, tea is being produced in India and it is being exported also, but how to confirm whether it is actually from Darjeeling or not? So that can be checked using isotopic fingerprints. Similarly, nitrogen. Nowadays, a lot of organic food, I mean, there is a great hype about the organic food, but apart from testing the pesticide residues, do we have other methods to check whether it is organically produced or not? So there is a method which uses nitrogen isotopic signatures, which tells that the fertilizers which have been used, they were from organic or they are chemically produced. So this way we can distinguish between organic food and the organically produced and the normal food. Then these are few examples which I have listed where isotopic signatures, they are finding application. Like natural versus synthetic product, then product authenticity. I have taken in next few slides only two examples. One is honey and the other is a flavor compound. Then geographical origin, as I have already told. For example, we have basmati rice from India and from Pakistan, so how to distinguish it. So high value items to distinguish the source. For example, my project partners at international level, they are working on coffee. Many people are working on coffee. Costa Rica coffee is very famous, it is a high value. Jamaican coffee is very famous. So how to distinguish? So that is based on these isotopic signatures. Then organic authenticity, source confirmation, chaptalization in wine. Wine is from fruit juices, but people are adding sugars for fermentation, which get converted into ethanol. So how to distinguish that? And in wine, this method is being used since 1990s. It is AOAC method, and now it has come for food products also. So I take the example of honey, which is finding application as a very good nutraceutical, I would say, because it has very good antibiotic properties as well as the nutrient properties. So honey adulterants, how we can test? Honey is being adulterated with foreign sugar syrups of different types. So what happens? Honey bee goes to the floral plants for collecting the nectar, for converting it into honey. And almost all the floral plants, they belong to C3 photosynthetic pathway. There are 3 photosynthetic pathways which are being followed by plants: C3, C4 and CAM pathway. So most of the floral plants, they are C3 origin, whereas all others, they are C4 origin. So honey will have the isotopic signatures of C3 plants. Here you will find that C3 plants, they have isotopic signature of -22 to -30 per mil. This is the unit of isotopic value, this is in per mil. And if corn sugar syrup, high fructose corn syrup, which is being used in pharmaceutical industry as well as in bakery industry, so that is also being used for adulteration, apart from the normal sucrose, which was very easy to detect by using the chemical methods. So if C4 sugar adulteration is there and we check the isotopic signatures of honey, the isotopic signatures will change from -22 to -30 range. It will be raised to -8 to -13. So which will indicate that it is adulterated with C4 sugar. But as we know that fraudsters, they are more smart as compared to the scientists who are working towards the regulatory side. So they came up with rice syrup. Rice is a C3 plant, so when you add rice syrup, will not be able to detect the adulteration. But again, the regulators are working towards that side. So another technique, EA-LC-IRMS came, in which we, first using the chromatography, we separate out all sugars, then isotopic signatures of individual sugars are analyzed. So likewise, I always say that there is a race between adulterer and the regulator, and which continues, and I think always the adulterer is little ahead of the regulator. So India is the first country, I would say, which has come up with the regulatory standards for honey for authenticity, because food quality, food authenticity, similarly natural products, quality and authenticity we are hearing since long, but this authenticity is a new concept which has come. So foreign oligosaccharides, C4 sugar content, delta C13 max, fructose minus glucose, protein minus honey, all these are isotopic signature values which FSSAI has come up with a regulatory framework for honey. So these are the equipment. IRMS is isotopic ratio mass spectrometer. We connect it with the front end elemental analyzer and liquid chromatography. So these are the techniques which are being used. For other purposes, GC-IRMS is being used. So another example I have taken vanillin, which is the flavoring compound. So vanillin is produced from the vanilla flowers as well as from the synthetic sources. Now how to distinguish it? In fact, for vanillin, there are two synthetic methods. One is chemically synthesized, other is biotechnologically synthesized. So when we check the isotopic ratios of these 3 different types of vanillin, so there was a difference in the values. So these values, like -22 per mil for natural and -37 to -36 for biotechnologically synthesized. Similarly for chemically synthesized, it is more negative than -25. So these isotopic signatures, they tell us whether it is natural or it is synthetically produced. Some other applications are like turmeric, curcumin which is isolated from turmeric, that is finding lot of application as antibiotic. So curcumin is also synthetically synthesized as well as from natural sources. We all know that from natural sources there are other components. We get a concoction which is always better as compared to a single compound which is prepared in the laboratory. Then saw palmetto, berries are with the medicinal properties. So these are also we can distinguish using isotopic signatures. Then squalene from shark liver oil and plant sources. So we find that vitamin D is from animal sources as well as from plant sources. So there is one concern about vegan diet also. People are, some people they don't want from non-vegetarian sources. So this is how we can distinguish whether that oil is from veg or non-veg. Similarly, research is going on the capsule shells which are gelatin. So gelatin has come from the plant sources or from animal sources that can be distinguished using this technology. So this technology has some challenges also. So those challenges can act as the opportunities for the researchers who are present here because this is based on database. All the analysis, we have to compare it with the database of the authentic samples. So database generation is very, very important. This is an area where researchers need to work. Then technical expertise in this area is still, I don't find much technical expertise is there. In India also in food for honey, now there are 8 laboratories, but when we started, we were the first one to start. In government sector, there are still 3 laboratories only. Then regulatory framework, because of lack of database, regulatory framework is not able to come up. So FSSAI also, we have, based on the published standard, we have given the limits, but still database generation is in process. Then authentic and traceable samples. We are working on honey authenticity, so we are finding it very challenging to get the authentic samples of honey to generate the database. So I hope I could complete in time. Thank you so much. Thank you.
Thank you so much, Dr. Dua, for helping us out for a few minutes.
No, I learned that Secretary Madam has come, so I try to finish
Thanks.
it in time.
Thanks a lot. And let us recognize Dr. Dua's contribution.
Thanks a lot for all the speakers for their more informative talk. Now it's time to honor our chairpersons. I would like to invite Professor Sarma and Professor Dulal Panda, Director. Please come on. Give the moment, please.
Check, check, check.
Good evening, everyone. All are requested to kindly remain seated for the valedictory function. ठीक लग रहे हैं? अब सामान कौन अरेंज